Hi, I'm Keisha, and I'll be talking with you about glucose 6-phosphate dehydrogenase, which is G6PD deficiency. Glucose 6-phosphate dehydrogenase deficiency. I'll be covering what is G6PD, what does G6PD deficiency look like, diagnostic procedures, the case study, the malaria research project, management and treatment of G6PD deficiency, and the gene tree that I worked on and the works cited. What is glucose 6-phosphate? It's an enzyme that helps the body convert carbohydrates and turn them into energy, and we just learned that in class. Responsible for the first step of the chemical pathway that converts glucose into the ribose 5-phosphate. It protects the red blood cells from potentially harmful byproducts that can accumulate when a person takes a certain medication or when the body is fighting an infection. There are 140 mutations identified with the gene G6PD. Mutations can lead to changes in amino acids that make up the glucose 6-phosphate dehydrogenase enzyme. These changes disrupt the normal structure and function of the enzyme or reduce the amount of enzyme in the cells. The red blood cells are unable to protect themselves from damaging effects of reactive oxygen. The damaged cells are likely to rupture and break down prematurely, and hemolysis is what it's called when they break down, and this is certain antibiotic infections and consumption of fava beans that can increase hemolysis in people with G6PD deficiency. And then I have this little illustration over here, infection, bacteria, certain medications and fava beans and blood cells popping. So how do you get G6PD? I am one of 400 million people who have it. My husband also has it, and I have 2 children. So instead of using the generic gene chart, I decided to do my own. So this is my son's father, who's XY, and this is me, and I have the R recessive gene, and this is my husband who has the R, and it's an X recessive gene, so it's only on the X, and it's passed from mother to son and father to daughter. So I have one of each. I have a son, Brendan, and I have a daughter, Kaylin. Well, my son's father did not have G6PD deficiency, so Brendan either has it or he doesn't. But Kaylin, being that both me and her father have it, she has G6PD deficiency. And of course, since daughters get it from their fathers, thanks, Dad. And it most likely in males of African, Mediterranean, Asian, and Middle Eastern descent, but they come to find that that's somewhat not true, because it is a X recessive gene. More women have it than men, and this is what the gene marker, what it looks like on the X, and then on the Y, because the Y is short and not active, it carries from the male. So diagnosing G6PD deficiency. It's called the fluorescent spot test. It's widely used in vitro, meaning blood draw diagnosis, and it uses whole blood, so you'd get a venipuncture at your doctor's office, or dried blood spots. So what they do is they have a reagent, and they take your blood, and they mix the reagent in your blood, and they take it out of the tube, and they put it on a filter paper, and they let it dry for 5 to 10 minutes, but I'll talk more about that in the next slide. So the fluorescent spot test was first developed by E. Butler in the 1960s, and it's sometimes called the Butler test. He was commissioned second lieutenant in the Army. He was assigned to work with the malaria research project with the Army, and he noted that these cells had more Heinz bodies than normal when treated with certain chemicals. So these are Heinz bodies. They're little protrusions on your red blood cells. So diagnosing it. So up here I have the blood and the reagent mix, and they're dropping it on the filter paper, and they put it in an incubator for 10 minutes. And what they're doing is they're looking for the reaction of the NADPH in the chemical process of turning glucose into energy. So then they, once it's dry, they put the spots under a UV light. And here's the UV light. And then if the NADPH fluorescence happens, then that means the G6PD is active and you have normal blood cells. But a lack of fluorescence signals that the G6PD is deficient. And I hope you can see it. I have it down here somewhere. What does G6PD look like? So if you have G6PD deficiency, that means your blood cells are destroyed and removed from the bloodstream way before their lifespan is over. So that makes you anemic. And with anemia, there's fatigue, dizziness, low blood pressure, rapid heart rate, because your spleen is like your blood filter. All the broken down and destroyed red blood cells go to your spleen, so your spleen will be enlarged because there's more deteriorated red blood cells. You'll have muscular weakness because those blood cells are premature and they can't carry oxygen to your lungs, to carry oxygen to get oxygen from your lungs, to carry oxygen to the rest of your body. And you'll be pale, you'll be cold, and some have signs of jaundice, means yellowing of the eyes and skin. And you'll be short of breath because your muscles and everything will not have oxygen to work. And in severe cases of anemia, you'll have chest pain, heart attack, fainting. Fauvism is also what G6PD deficiency is called, because in severe cases, it can send you into a hemolytic crisis, and you'll have to go to the emergency room, get a blood transfusion. But it's accelerated by the consumption of fava beans, hence Fauvism. This is the malaria research project in the Army. I found out I was G6PD deficient when I went into the military. They did a blood test and told me that I was G6PD deficient, and I could not take the malaria pills. And we get deployed often to regions where Middle East, Africa, regions where malaria is prevalent. So being that I couldn't take the malaria pill, I was kind of exposed to malaria. If I was exposed to malaria, then I would have to come home, of course, and be treated. But in this research project, they found that it was first recognized in African American males who developed acute hemolytic anemia after undergoing therapy with primaquine, which is the malaria drug. And they found that their red blood cells had become rigid and non-deformable, and they pretty much were undergoing severe hemolysis. And the normal half-life of a red blood cell is 62 days, but they found that the half-life decreased to 13 days in G6PD deficient people. So when it was introduced to the malaria drug, and initiating the malaria therapy, the hemoglobin typically falls from normal in males is 12 normal, and it went down 3 to 4 grams per deciliter. They had a sudden onset of jaundice, pallor, their urine was dark, and with peripheral blood smears they showed evidence of hemolysis. And once you take away the agent that has caused it, if it's infection, fight the infection, or if it's the malaria drug, take the drug. out of the plague. They usually return to normal. So, fun fact about malaria: the malaria parasite cannot attack in an environment with premature red blood cells. So people in with G6PD deficiency have premature red blood cells, and hence they have been shown to show decreased level of activity when malaria parasite is present. G6PD deficiency management and treatment. Avoid the oxidative stressors, the anti-malarial pills, sulfonamides, aspirin, fava beans, peanuts, blueberries, they said. The treatment is remove the cause of a hemolytic crisis. In severe cases, you need a blood transfusion, and a splenectomy, which is removing your spleen, is not generally recommended, because if you have it, where will all those blood cells go and get destroyed? And folic acid and iron are potentially useful in hemolysis and anemia. And this is my gene tree. This is all primates, because there was no other tests on any other animals or any other species. And this is me and my baby Caitlin, and a chimp, and an orangutan, and a gorilla, and these wonderful monkeys. They're so cute. And that's all. That's G6PD deficiency in a nutshell. Thank you.